It's nice to get an applause before I've said anything, so thank you. So thank you all for bearing with us until the end of the day. Thank you to the organizers for the invite and for pulling together such an amazing panel. I don't think we need introductions from these individuals. I was just talking to Phil and saying I wonder what the average age of the audience is and what the average age of the panel up here is, and I won't tell you what Phil said, but but what I thought we'd do just very quickly is just quickly go through each of the panelists and just ask them to give us just in a couple of minutes what their perspective is of the gap between academia and industry. Is it big? Is it getting bigger? Is it getting smaller? Whatever, etc. So, Phil, why don't you kick off?
Okay, well, I don't think I'm going to say that because I can't really tell whether it's getting bigger or smaller, but let me just get the discussion going, just tell you how I've tried to plug a couple of gaps over the years. So for those of you who don't know me, I've been working for nearly 50 years now to understand the role of protein phosphorylation in cell regulation and human disease. For the first 25 years, no company had the slightest interest in anything I was doing. And then suddenly in the mid-1990s, protein kinases became an emerging area of drug discovery. And I wondered, how I could help bring out the expertise that I had in my unit to bear on this problem. And also because it was becoming clear that you could actually develop specific inhibitors of these kinases, I realized that these would be terrific reagents for my own research. So I set about something that took about 3 years to put into place. It's something we call the Division of Signal Transduction Therapy. It's as I say, it's a collaboration was to try and help pharma through the early stages of emerging drug discovery, how to allow them to initiate a large number of drug discovery programs, because there's 500 protein kinases encoded in the human genome, and also, and perhaps more unusually in academia, how to develop the critical mass of reagents, assays, and technologies which would help them to get their programs off the ground much more rapidly, because this was at a time when these reagents weren't available commercially. So, I think, in my experience, what pharma need in this area is a critical mass of expertise in the area they're trying to learn. If they can get it in one place or even 2 places, this is a terrific bonus because they don't have to chase around to 20 different labs getting the expertise. So in that side of things, I was lucky because in 1990, the MRC had set up a special unit under my direction, and so we were developing very rapidly a critical mass of expertise with many principal investigators working on this topic. And then when we actually got this in place in 1998, we were able to use a lot of the money that we got from pharma to put in the other critical component in place, which was this critical mass of reagents, assays, and technologies. And I think this has turned out to be sort of very beneficial in keeping this collaboration going. Miraculously, it's still going 20 years later. It's always been with 5 or 6 different major pharmaceutical companies. So these are really the critical aspects. You need critical mass of expertise, critical mass of assays, reagents, and technologies, but you need multiple labs in academia working together in a coordinated fashion in order to be able to deliver to pharma, what it needs in this area. So I think the way we've put this together is generally applicable because we've done it a second time. In about 10 years later, I became very interested in another emerging area of drug discovery, which is protein ubiquitination. And I managed to persuade the then Scottish First Minister, Alex Salmond, to give me quite a substantial amount of money to set up a critical mass of principal investigators working in this area, and also sufficient money to again roll out saturation coverage of all the reagents, assays, and technologies that are needed in the area. And this has turned out to be sort of equally useful, and it's probably prolonged the life of our consortium with pharma by about another 10 years. So I think this is a generally applicable thing. So you not only need a critical mass of expertise so that pharma can get nearly all they need in one place, but you also have, at the same time, have to get a critical mass of reagents and technologies at least together, at least until companies that can produce and sell these reagents in the large amounts needed come off the ground. And actually, quite a surprise to me is that I've never set up a spin-out company, but it turns out I've been very successful in developing spin-in companies, because every time we develop these critical mass of reagents, companies appear from nowhere at my doorsteps wanting to sell them. And so we've actually had, we had a company called Upstate that actually took our kinase profiling technology, because there's 500 kinases. Companies want to know, does my drug inhibit one of the kinases or all the 500? So to do this, we obviously need huge numbers of kinases and assays. And then this company set up in Dundee. It was very successful. It went from 0 to 125 employees within 5 years, and then it actually was sold for a lot of money. And actually, we used the royalties from that money to actually build a new building in our institute. So, and then similar thing happened with ubiquitination. A company called StemGent said that they would like to come and do something similar, and they would call the company Ubiquigent. And this is, again, starting to do quite well. And so this has been a big surprise to me. The other big surprise was that by interacting with large numbers of pharma companies at the same time, who came to our place very frequently, it got a lot of our students and postdocs very interested in actually joining companies, which wasn't the case before. So I think before we started this collaboration, the number of students and postdocs in our unit who joined pharma was vanishingly small, but now they can see the tremendous challenges and interesting things that you can do in pharma partly as a result of our collaboration, and this has been very helpful as well. So so I think it's partly been closed in some areas, so I still think this is a gap, but hopefully less of a gap in the areas I work with.
Phil, thank you very much. So critical mass, talk to politicians, and spin-ins. Jane.
So I guess I'm the industry
and you're the youngest member by far of the panel.
Thank you, yes, I hope. I think the first thing to say is that industry isn't binary. I think we've had a lot of discussion around what's names and binary sort of definitions, and quite a lot of people, well, most people from industry start out in academia, as I did. I worked as a postdoc. I went to a small biotech with Greg in the sort of early 90s, and have gone through various mergers and acquisitions. So I've seen the whole spectrum of academia right through to big pharma, and I'm now officially part of big pharma. So MedImmune is the biologics unit of AstraZeneca, and we're very involved in checkpoint inhibition and antibody therapies. So has the gap, the question was the gap, I think the gap has closed a huge amount in the 25 years that I've been in industry, and I think that gap's closed because of a lot more openness around information sharing, genuine collaborative behaviour from industry, and a genuine interest in driving science forward. And I think that's a very, very positive place, and I see that continuing. I'd say there still are gaps that we need to sort of think about how we address. And for me, I think the three areas that I would call out is, first off, really helping guide the academic community to have a little bit more commercial awareness as to how to position some of the ideas and technologies that are coming through. I think the second is around making time. I think we're very good at connecting at a grassroots level, and we're quite good at connecting at a strategic level across industry and academia. The PIs who are have a very high workload, and our managers who have a very high workload need to make more connections. So I think we need to sort of try and think of ways of closing that gap. And then I think the final one is, I think there still is a funding gap I also am chair of the Bioindustry Association, which is a trade association for UK biotech. I think we're quite good at starting companies, very good at starting companies. There's some great examples of how we're growing some of those companies. I think we still need to think about how we grow those companies to fully get the benefits of translation.
Jane, beautifully succinct. Thank you very much. Ray, are you going to give us a USA perspective?
Well,
I keep hearing the USA is better than the UK.
I think you should remember that practically the great majority of innovative science takes place in universities. And they start there, most of them. Very few times you can say, oh, Merck did this, or AstraZeneca did this. It always starts somewhere in university. So I recommend the young people here to actually stay in universities because you have a lot more freedom and nobody's going to fire you. So that's the advantage. So I never left academia. I've been at Emory University before. I was at Yale, 2.5-year postdocs, and I went to Emory University, and I've been at Emory this year. I'll celebrate my 40th year. But I started many companies, as some of you know. Some successful, some very, very successful by any standard, and they're probably going to write a law review at Harvard, business review at Harvard, or something about what we've done, been able to accomplish. So I think the gaps are narrowing because usually the companies come. You got to advertise yourself. You got to learn how to become a PR person. You need to go to meetings. You need to talk about your stuff and not hide it. File your patents and then go ahead and show what the goods you have, because nobody's going to know about it if you're too secretive. That's one of the biggest problems in communicating with industry. But then industry has to take the hook, and sometimes they don't, and that's why you have to decide to gather your friends and colleagues and people smarter than you are and form a company, and that's what I've done. You cannot do it on your own. That's pretty obvious. You need to do startups, and then you need to entice the big pharmas to come and buy you out. That's the best outcome, I think, unless you become another Gilead, or you become an AstraZeneca. But these are dying dinosaurs. They have a lot of money, but not much innovation right now. I think the innovation, the real innovation is from universities. Just remember that. So I'll stop there.
My goodness, that was a
You can respond
comment to finish on, wasn't it? So I was thinking before this panel, I mean, if you think about in this country, truly successful academics, created biotechs, created a lot of impact for patients, real entrepreneurial flair, etc. I couldn't think of anybody better than Greg, actually. And I like Greg's preface to this meeting. I mean, he talked about sort of science and technology, but he also talked about sort of politics and business and marketing, and Ray's just talked about PR. So, Greg, give us a few words of wisdom. Why are you so successful?
I couldn't possibly give that secret away. Going to the question of a gap. The sort of gap's a multi-dimensional gap, as has been implied. There's not a single gap. There's lots of gaps. And so at some point, if you're a scientist and you've got a discovery, in my case it was technology is what I've been responsible for spinning out or in some way getting industry to use. There are a number of kind of things you're going to need to put in place, you or the industry is going to need to put in place to get that successfully used to make a therapeutic, in this certainly case I'm thinking about. The first is you better make sure your technology works, right? So it's really got to work in some way, and you need to have patents. You need to have some key proof of concepts, whether it's cells, animals, most preferably people, if you can do that. You've got to have a business plan, really, or someone has to have a business plan. And you've got to get funds. So all of those elements have to, and probably more as well, have to come together. And then you say, well, okay, so it may be a bit depressing if you think you've got to solve all of those problems yourself. Because actually there are other industry or other people may be able to help you deal with some of those. And I can illustrate this by the first example of commercialization that I was involved with was developing of a technology for humanizing antibodies. So this was pure genetic engineering. We filed patents. We did proof of concept in animals with other fellow academics. In Cambridge with the NHS, we showed a humanized antibody in a patient. And we got all of that done very quickly. And with that, essentially with all of those elements assembled, we were able to interest industry to license the patents and to pick it up. They could see there was something that could work, the patent position was clear, they could run off and do it. And that gave rise to, in many ways, the most successful thing I did, which is the humanizing of antibodies. There are, there's Herceptin, Avastin, Keytruda. Many of these big blockbusters come from that humanizing technology. And that didn't really require me to get involved in raising money. What it did require us to do was to adopt an intelligent and easy licensing policy. So we followed the Stanford model of Cohen Boyer, where you have low royalties, a modest upfront payment to get people in, get them started, make it available. So that is one approach, and actually the licensing approach is often one of the best ways of getting things out there, but you may have to do quite a lot yourself by way of proof of concept going into patients, etc., to be able to get people to take it seriously. So we didn't get licensed to it to one particular company, it got licensed to all companies simultaneously. The other approach that I took, which will maybe give you some comfort, is all of the things I've described, like technology, patents, business plans, proof of concept, and funds, were not in place at the time we started the company. And that was the development of the phage display approach, or creation of antibody repertoires and phage display, which in the end created Humira, the world's leading pharmaceutical drug, or it was last year anyway. So really where we started from there was just the notion of creating large repertoires of antibodies. And at the point we founded the company, we had no proof of concept other than generation of repertoires. And they weren't really validated. We just did some sequences and showed they were different. We claimed there were billions of different things in there, but we didn't actually know for sure. The next thing we knew we had to create was a way of selecting those which had binding activities to the particular target, because that's an element of it. And in fact, in setting the company up, that was the only plan we had at that point. We had no idea what markets were. We decided we just have to solve the problem of selection. So we employed a chap, John McCafferty, who was one of the first employees of Cambridge Antibody Technology, and he came to my lab and he did it there. We tried a couple of approaches, but that was one of them, and that worked. So therefore, in fact, we didn't even have the technology in place that became the cornerstone at the time we founded the company. We had patents only covering one of the technologies. We had no proof of concept. We had no human antibody created at all actually for probably 2 or 3 years using this approach. Our business plans were really all over the place. We tried to work with, the board wanted us to work as a service model. The company management wanted us to create our own products, but didn't know what to create them to. So, actually, that was all. And we had very little money. So we had a small amount of money from an Australian horse racing company, which was the only place I could get money from. And they saw this as being like a bet on a horse, not an investment. So actually, everything there, wasn't right in terms of the gap. But what we had, and what I'd learned with humanizing, was momentum. Actually, sometimes by moving fast and quickly delivering on this and that, you keep the momentum, keep the interest, and I think that's an additional thing. You could shoot across that gap if you've got enough jet power behind you, and you may be able to fit the other things into place as you go along. So I'll leave it there.
I won't ask what Greg's connections are with Australian horse racing. But what was interesting listening to Rick's talk earlier on, he talked about, what came across in his talk was his passion for science and his humility. I mean, it's such an inspiration. But he said at one point, he had loads of scientific questions, and that's why he set up a company. Many of us think we need to have a plan in place with clear milestones, and then we go to funders and we set up a company because we want to translate ideas. But Greg's just been talking about, he had a platform, he wasn't quite sure where it was going to go, but he still set up a company. It's interesting. Stephen, whenever I see you, everybody's usually groveling to you for money. So Stephen sits on a pot of 100 million at the Wellcome, and he doesn't know how to spend it, so ask him. So.
Yeah, when you join the world of being a funder, they say two things happen. The first is you never have a bad meal again, and the second is you never tell a bad joke. So I'm expecting laughter there. It's pretty hard now to come up with anything particularly insightful, which is my default setting anyway, not to be able to do that. But a couple of observations. The first is, if you're looking for the right answer, guess what? It doesn't really exist. There is no right answer machine. One of the things I've learned in working at the Wellcome Trust, where we've been trying to think about what's the big thing we should really work on, that we should try and fund? And the answer is that you can't boil this down in an engineering or a scientific way into, this is the right answer, or that's the right answer. You just have to try something. And I was very struck by Stephen Friend today talking about sometimes just simply taking the next step being important. And I think we heard that from Greg. Have an idea, have some form of vision that something might be useful, and then taking the next step. So I think that those are important. And also, don't forget, and maybe the age of the panel will tell you, it usually takes about 20 years to be an overnight success. So the gaps that I think exist that are material for translation, one of them that worries me a great deal is actually the age of people at which they start to translate. Very often people, and it's something that is particular in the UK, that people get an academic appointment and their first academic appointment is getting later. They get their first independent grant and that's a bit later. They get tenure, that's getting a bit later. And so suddenly they're in their mid to late 40s before they feel that they can really take a big risk on setting up a company. And I think that's something that we have to really try and do something about. So one of the gaps that I think is missing is to focus a bit too much on academic faculty as entrepreneurs. Of course, it's very important that academics create the environment in which translation can thrive and lead by example, but it's also important that we encourage people as early as possible to think about setting up companies, about working with industry in a multitude of different ways. And I think the final thing that I would add is I think there is a danger of us missing out on what's happening in other disciplines. There's a bit of a danger of the biotechnology and the biomedical world becoming a little bit too insular and not thinking and learning enough from the speed and agility that is happening in other sectors, whether or not that's in the automotive industry, whether or not it's in the various engineering sectors. And it's still, of course, there's collaboration with many of those disciplines with biomedical scientists, but we're not learning about the speed and the idea that it could take, for example, in a neglected indication, it could take 18 years to go from an idea to something that actually makes it to patients. It's frankly a failing of us all in harnessing our intrinsic creativity to get there faster. And I think that there are many disciplines that actually are showing us that we must get there faster. So cross-disciplinarity and youth would be my two words.
Thank you, Steve. You know, it reminds me of a couple of years ago in Oxford, we set up a Dementia Institute, and Jackie Charlton, you know, the motor racing driver, he came to see us, and at the time, Jackie Stewart, sorry, I beg your pardon, he set up a charity called Race for Dementia, because his wife has dementia. And he was saying that he wants to fund young kids. He doesn't want to fund old fuddy-duddies because they're too set in their ways, and he wants new ideas, he wants sort of crazy ideas, etc. And secondly, about 8 years ago in Oxford, we did an innovation review, and we had somebody over from Stanford, and he was saying, On the West Coast, most of the companies that are being set up, they're being set up by these 22, 23-year-olds, not by faculty members, which I thought was an interesting comment. Anyway, so we've had a bit of a perspective from sort of academics, industrial scientists, entrepreneurial scientists, a funding organization. I know the organizers wanted to sort of keep maximum time for questions, so let's just go straight into questions. There's a question right over there. And make them hard because the panel's tough.
Hi, my name is Peter Todorov. I'm coming from Harvard Medical School's Lab of Systems Pharmacology. I'm somebody who has a background in both the biology and currently I develop software. So I'm well aware of the different pace in both sides of that. Do you think this is directed at Professor Caddick and the rest of the panel, do you guys think there's an inherent limit to the speed that biology and biotech can move at? We have to wait for cells to grow, whereas I can just push my code and make a website very quickly. So are there inherent things that slow us down? Thank you.
Ray.
I could quickly answer this. I mean, I think looking at, for example, our discovery of the cure, the so-called cure for hepatitis C, technology was extremely important. If we didn't have that technology, we wouldn't be able to do what we did. And the two technologies that turned everything around was real-time PCR, which in the early 90s was relatively new, and the machines were exorbitantly expensive. And then, of course, the replicon system, which became available, and that really changed the dynamics. Instead of using, surrogate system, we could actually use a real HCV system, although it didn't produce virions, and that was a key turning point for company Pharmaset. Pharmaset discovered the drug sofosbuvir, which actually overtook, for at least a few years, Humira, and now it's dropping down, of course, a little bit because we cure people. That's the difference. Humira you have to give forever, so that's one of the problems. So clearly, this technology coming at the right time really helped. On the other hand, imagine if HIV occurred in the 40s. We'd all have HIV in this room, probably. I mean, it's amazing how timing is everything.
Don't you love the competition amongst the panelists? Stephen, did you have any comments to that?
Yeah, I mean, it's a complicated question, but let's not forget what we've already seen about the way that science can move. So access to scientific information, which is really the lifeblood of great ideas, creativity, and ultimately solutions. We've seen that revolutionized in 20 years or so. The press of a button, I can get access to really some of the most up-to-date information. Technology will also revolutionize. We will see in our lifetime, I'm sure, at least a couple of things. The first is the ability to get a structure of all proteins of a proteome within days or weeks. And I think next generation sequencing will become so routine that it will be like taking someone's temperature. So if you imagine what could then happen, how variations in the genome, post-translationally modified proteome, could tell us about disease and living healthily with disease, as we heard about from Stephen Friend, then I think that opens up the imagination for us to make progress faster. There's no doubt that product development takes time, and thoughtful and ethical clinical evaluation of products should not be, there should be speed but not haste.
And I think if I could just add to that as well, I think in terms of the technologies and the techniques, absolutely there's no reason why biology can't move at the same pace. And we see CRISPR, we see fantastic improvements in productivity and ability to get answers. I think the point that Steve just made around the regulatory environment really is the thing that takes the time in terms of drug development. And that's an area where we do have to sort of have a really serious balance. We need to be regulated, and we need to take that very seriously. We need to understand that we mustn't put people at risk, and getting those risk benefits correct in a way that delivers maximum benefits to patients without the risk is the challenge. I think the great opportunity we have at the minute is that there are a lot of people from more of the digital community, the IT communities, asking questions about why the regulations are as they are. And I think there are very good reasons for that, but having those questions asked and really sort of pushing at the boundaries of that is probably what we should be doing. And I think everyone in this room can sort of think about how we can contribute to that.
So code alone isn't going to cure all disease. So, another question over there.
My question was that from academia to industry, is there a middle gap? Because in my personal case, I went straight from academia to industry, the manufacturing industry. And I'm not talking only pharma industry or drug industry, and you have a lot of research itself in the industries. So a lot of the academia go into R&D in the companies. And the companies today, like BW, have resources which are far in excess to academia. So that is one area. Can the panel tell us something about startups? I'm noticing today worldwide more and more youngsters are coming into startup companies, and maybe people here can tell us something about the startup.
I mean, there's no doubt that academia is not well placed or necessarily skilled to do manufacturing or toxicology or scale up or whatever. Exactly. But in terms of startups, Greg, do you have any comments?
Well, I was going to say my story on this would be in the early days of working with antibodies, the general view from the pharmaceutical industry would be those will never be pharmaceuticals. And that was because the industry was largely at the higher levels populated by chemists who actually thought of this as a biologic we're looking at. Actually, it's not a proper pharmaceutical. Now, you refer to their labs being full of lots of power in their labs, and it's true they do, but they have a hierarchy which actually is probably several generations out. So we found people in large pharma when we were doing our antibody work in Cambridge Antibody who were desperate to do something with antibodies, but the guys high up didn't really want it because they didn't take it seriously. They thought it was playing. And I think the same is true with IT. Essentially, from the existing industry leaders, it's very difficult to get a paradigm shift, and that's why startups come, because people look at something from a completely different perspective, and then you can get real innovation. So I think the power that pharma companies have in terms of being able to fund things if they want to is attenuated by the fact that usually they don't want to do anything that steps too far from their traditional patents. And indeed, committees make decisions there. So usually, any decision made by a committee, it's the same as grants in academia. Any decision made by a committee will be safe and not likely to be sort of revolutionary.
I would say I think that is changing quite significantly.
AstraZeneca, I'm quite sure.
No, not just but I think we're sort of going back to the sort of how you get disruption.
Yeah.
We used to play, I don't know if you knew, Greg, we used to play the game of who owns the zebra in terms of who actually holds the funding decision to try and get a technology or an asset brought into a larger company. And actually, that's one advice I would give actually the people in this room is do try and work out who the decision maker is in a larger company around adoption of technology, because sometimes it is a committee, sometimes it is actually a champion, and understanding how you get through the gatekeepers to get to that champion is actually really important in getting really good adoption of ideas.
I agree with the questioner that I think that academics in industry are on the ascendancy. Shown very, probably most notably by Genentech, but lots of other companies like AstraZeneca have their own postdoc and PhD programs. So I think this is going to have quite an impact
But we don't want academia to become industry, and we don't want industry to become academia. We need to play to each other's strengths. Stephen had a comment.
Yeah, so so out of quite a lot of what you'll hear, I think there's one thing to remember, which is that always be worried that if you've got an idea and everyone tells you it's a really good idea, then I have a saying that in my team, if we all agree, then we're probably all wrong, because consensus usually is conservative, and that's sort of Greg's point. The decision making is really important. So, and the problem when you're starting a company or you're starting to embark upon any problem which is really difficult is that people are, if you're working on something that's hard enough and important enough, almost everyone's going to tell you that you're wrong. So you have to have resilience and persistence in order to get through that. Because it's fine once you've succeeded, like people on this panel, but when you're going through it, it's actually, it can have its low points as well, and you have to be persistent.
And then the short question for me. That is the question of...
No, I think we should spread the questions out a bit. I think Ray wanted to follow up.
Yeah, I think academia is, talking from academic perspective, I mean, we have the NIH funding. We get $500,000 for individual drug. You have a great idea, you submit a grant, and you believe in it. You get rejected usually the first time, the second time, maybe the third time you get it. And clearly it sharpens the mind, and you are in charge of your own destiny. If you believe in it, you keep going. And I think it can do things with $500,000 that would take industry several millions to do. So we can be a lot more efficient initially to get to the certain point to start up a company or license your technology, at least file the patent and move forward.
So more questions. Just here, second row, middle. I'm going to see one of Stephen Caddick's committees at the end of this month groveling for money. So I'll remind him that decision-making by committee is mediocrity.
Actually, my question is pretty related to money, and it's also a different perspective. I come from Mexico. I'm Buenos Aires. I'm genomic biotechnologist. I'm in a certain part of my life, I was forced to continue doing my master's because when I graduated, I could not get entered to the industry. Because the offer that I received from the industry in my country, a developing country, many times is really, it's a big deception. When they're offering to pay you less than £200 per month, when you got a degree of biotechnology. And there's a different phase of what we need to encounter in developing countries. Many times biotechnology in our countries is still a new word that produces uncertainty for the industry in our countries. Despite that many big companies know which is that. But the human resources team in the branches, they don't know. So that gap creates another gap that we need to face. If we could not provide a successful entrepreneurship project because of governments and not really providing the opportunities to get, the spaces to get that entrepreneurship. So we got two options. Or stay in our countries, forced to be just in academia, that pays almost the same, but provides better professional opportunities, or leave our countries and never come back. And that is a problem in our country. Actually, Mexico is quite a funny case, because I could say it specifically.
So what's the question, briefly?
So what we could do in those cases, when our reality is not a developed country,
A quick
what we do to move academia and industry in that reality.
There's no doubt that we in the UK, especially sitting in this town, or the US, we have it really lucky, and there are major, major challenges. Any quick answers to that?
The cost of living in Mexico is very low, so you don't have to worry too much about money. And in fact, if you're going to do science, you really shouldn't worry about the money. You should worry about doing science for the sake of science and developing something. And when you have something, form your own company in Mexico. That's my advice to you. You'll be very successful. Look at it. Look at Spain 30 years ago. There was hardly any biotech or even pharmaceutical companies in Spain except generic. Look what's happened in Spain recently. So things change, and even we consider the poor European country, but it's no longer.
More questions?
So I'm Srinath. I'm a third-year undergraduate student from the Institute of Technology, Madras. And I am in a real dilemma about transitioning from academia and industry. And I've got the best time here to answer my questions today. So, first of all, I have two questions I'd like to get a combined answer maybe. When is it the right time to transit? So, do people have to go ahead with a PhD to get more feel into what really academia is, and then maybe move to the industry, or can they go ahead after their masters? This is one question. The second one is, how would you decide between research in academia and research in industry? What are people looking for, and what traits do they have to pursue research in academia, and what traits do they need to have to pursue research in industry? And what would be the role of incubators in this?
Okay, quick answers.
Yeah, quick answer. I think clearly the academic part is important, what you just said. But I think to do the transition, you really need to establish yourself as an investigator in academia. Have papers, have quality papers, at least, and sometimes, people jump out when they don't get tenure, but it doesn't really matter. Life goes on, you'll be successful irrespective of tenure or no tenure. But I think the important thing is to establish yourself as a good scientist, reputable, where you can reproduce your data, the people will believe you, and you'll be credible when you go in front of venture capitalists, when you go in front of bankers or hedge fund managers or whatever. You'll have the opportunity to express. They'll see your paper in quality journals, and they'll take you seriously.
Any wisdom, Greg?
Well, I was going to say that the point of a PhD, can be to get papers, but it's also to confront you with a problem that you've got to solve rigorously. And actually, you'd better go through that, because you might think you've got, with an MPhil or actually a first degree, you think you're quite clever, but actually, until you've actually confronted an intractable problem, which your PhD is, usually throws up, right at the frontiers of knowledge, then frankly, you've got no idea of exactly how you will deal with the problems you will get thrown up in business. So I think you have to have a PhD for recognition, but also just to make sure that you are capable of dealing with something at the level at which you're going to be expected to deal with it in a company. Of course, if the company has nothing to do with science, and actually it might be some marketing thing, some Facebook based on patents, those things you may not need to have a PhD, but I would still advise it.
I agree. We need to learn how to ask questions. We need to learn to be resilient. We need to learn to communicate. And in your PhD, you do that. I can tell you from personal experience, I did a postdoc in Oxford, and I was an arrogant shit. I thought I knew it all. I went into industry, and frankly, it really knocked humility into me. I had no idea about getting molecules across the blood-brain barrier. I had no idea about toxicology. I had no idea about how most of the animal models were a waste of time, etc. You learn so much. I would encourage everybody to spend time in industry as well as academia. I think it's good to learn each other's perspective. That way you trust each other, you build relations, you grow your network. And that's what I see happening in the US. In the US, you get people like, we've got some exceptional individuals here, and there's always exceptions. But in the US, I see people, they're working in Harvard, and then they'll go off and work in Novartis, and then they'll set up a company, and then they go back to MIT or something. And that moving around, I think, is critical for building an ecosystem like they've got in Boston.
I agree with everything the panel has said, but I would also say that if you want to go into industry, I wouldn't leave it too late. I don't think you want to be well into a second postdoc before you start thinking about it.
I'd actually agree with that. I completely agree on the resilience point. I think for a PhD does give you that discipline and resilience. And when you look at the people in more senior positions in our industry, it's very unusual not to have a PhD, although there are lots of exceptions. But I think, again, industry does provide a really, really good training ground for technical disciplines, but also for really sort of understanding how you actually drive to a specific answer to a challenge, and you have that sort of community of sort of care to drive that as well, which I think is a very useful experience. We have a lot of moving from industry back to academia and back again, and I think as Chaz just said, if you can move around, there are fewer barriers, and I think that's a gap that's changed in industry. It's a lot easier to go in and out of academia and industry and have those sort of sabbaticals and changes of direction. So.
I mean, Phil's just got another 2 program grants, but if it wasn't for that, he was thinking of moving into MedImmune.
Well,
to work in my daughter's team.
Yes.
Yeah.
But be careful which lab you do your PhD in. You know, go and work in a lab that's got international profile, doing cutting edge science, and a supervisor who's going to really give you a hard time. Don't go for somebody who's just going to wipe your ass every day. Question there.
Hi, great panel. Very much appreciated. I think a lot of people in this room are interested in entrepreneurship. You were specifically mentioning that out in San Francisco, 20 and 30-year-olds are starting companies. But biotech is a fundamentally experience-driven industry, navigating regulatory landscape, clinical trials, etc. And most VC-backed founders are 40 years old or older. Do you think this will change over time, and do you think there's any market for younger people in the 20s and 30s to start disrupting the industry?
Stephen's nodding his head.
Yeah, I think so. I think it's coming. I think it's... every other industry is changing. The industry that has changed least is pharma and biotech, by comparison with all other industries. I'm not talking about the technologies which have been breathtaking. We've heard about some of them today. But the underpinning business model of pharma remains fundamentally the same. And that's not really remained the same in almost every other sphere of human endeavor. So once the business model changes, which is, for example, where do you extract value as an investor in pharma? Do you do it at the point at which you sell a drug? Do you have a subscription model? Do you use healthcare? Do you think about lifelong brand association? Whatever it might be. And those questions will be asked by people who are probably earlier in their career with less to lose.
Ray, quickly.
Yeah, a good example you should read about this person is Vivek Ramaswamy from Roivant. He actually worked for a hedge fund when he was in his 20s. And by the time he was 30, he formed Roivant, which he basically got a recycled drug from Glaxo, and built a company around that and raised a tremendous amount of money. Now he's formed several companies. He's now 32 years old, extremely successful, and you'll hear more about him. He has great ambition. So there are exceptional people out there. He did go while he was working as a hedge fund manager. He was also doing studies. He didn't waste time. He actually went to Harvard, did an MBA, and then went to Yale and did a law degree. So he has a law degree from from Yale and one from MBA from Harvard. And now he's setting up his own company. So you see, you have to work hard and study while you're doing this. And clearly you can do it.
I think the important thing is just make sure you enjoy what you're doing. Don't just focus on making money, you know. And you guys are all going to live to 100 anyway. You could have 2 or 3 different careers. So I wouldn't worry about it. So more questions. Right at the back there, please.
Hi, thank you so much. Yes. So we hear about many successful stories clearly, and those are the ones that get publicized, and those are the ones that we strive for. However, I'd love to hear a little bit about how when early career academics, such as PhDs and postdocs, are looking for their next step, they look often for other things and elements in their life, and not just that success story, but maybe some certainty in their jobs in the future. So for instance, a postdoc looking at opportunities for grants and such, they maybe get certainties of 3 years ahead, or maybe 2 years ahead with funding bodies. And maybe we talk about how this research can be a lot more cutting edge than that which is happening in industry, but it is still regulated by whatever council that is deciding what kind of research they're going to fund. So not all the crazy ventures actually get around. So how do we think about when looking at opportunities to take, whether in academia or industry, when you're looking for not that uncertainty that in your 30s you're going to be scrambling for finding your next position, which can often take a toll in many other pieces of your personal life. So maybe that discussion of mental health is what I kind of want to bring up.
Phil, Greg, tell us about your failures. You must have had some failures.
I
I don't know about the mental health.
Greg's failure.
What I could say though is, look, it's true. If you wanted to be, as a postdoc, you're quite right. The academic life is very hard and it's short term. But in fact, what a number of people have done is to spin off, for example, work that's going on in the lab that may be, work that's actually partly their mentors and partly an angle on the project they've got, and leave. So, for example, I had actually he was a postdoc and then he became a junior group leader, but only briefly, because actually we had been working on antibody domains, Ian Tomlinson, and he thought it was so exciting we should set up a company on it. So actually it was sort of joint work that had worked over a number of years, and he took the initiative to spin it off, and we worked together to achieve that and set up a company called Demantis, which worked very well. So that gave him his opportunity to do that, even though, in fact, it wasn't something that he completely and utterly originated with him. You don't have, you may be things around which you can take, or part of the project which you can develop, the PI that you're working under may not mind you taking that.
My first research grant was turned down, my first application.
I'm glad to know that.
The reasons were, this is in 1972, the protein phosphorylation, that everything was solved. We had an example of this already in the field of glycogen metabolism. There was actually only one kinase at the time. The second reason was, well, even if it's not all solved, the other referee says, how can you compete with the two other big groups working in this area out there? So basically, these are the things that every young person will have to, to confront when they apply for their first research grant. How can you compete with the people who are already in the area? And, isn't everything solved already? So I'm afraid it's something you'll just have to live with.
Yeah.
And you just have to keep writing more grants.
I think you need to sort of think, what do you mean by certainty as well? So, I think, I've been in a position of having a postdoc coming to an end and then moving into a biotech environment, not actually realising how unstable a biotech environment could be, Greg. So I think at one point we had 3 weeks' money in the bank.
That's right. But I'll tell you, biotech isn't as unstable as large pharma.
Well, exactly. So exactly the point I was going to make was that the great thing about being in biotech was we were doing something we really believed in and we loved, and the science was great. And you can do that in big pharma as well, but you actually have less control over what you might end up taking forward. Decisions can be made for very good strategic reasons that don't give you certainty. So I think the key is really just, as Chaz said earlier, focus on doing the thing that you're enjoying and that you're learning from, and have self-confidence that, if you go through some kind of uncertainty, as long as you've learnt from your experience, you will then be in a great position to do something different. And we've all gone through huge numbers of failed projects through our careers.
You have to be very persistent and very resilient.
Be resilient, but just make sure that you're always learning and that you use that learning in the next thing.
I think that those are important comments, actually. So that if you're passionate about something and you own it and you're controlling your destiny, it shouldn't be stressful and no risk of mental health. If that. Ray.
So I think I may be controversial, as I usually am, but clearly, if you're in academia and you got a great product and you can get license it. That's my advice to you. If you cannot license it, then form your startup and find a way to do it. It's not, you have to believe in it. You have to go with your hat and ask money from a lot of people. Be careful of lawyers and other people like that who will take all the money that you have. Make sure the money is spent wisely and the money is on target, meaning every experiment should count towards NDA approval. That's what you should do, and that's my advice to you.
I think the other thing I would say is, life in industry, big pharma, isn't all rosy. The sorts of people who work in big pharma are actually pretty much the same as the people who work in academia. There's a few differences, etc., but largely they're bright people, they're ambitious people, they're hardworking, they're committed, they want to deliver new medicines, etc. So I personally believe there isn't that much difference between the scientists. But, you know, working in pharma, there are lots of committees and you don't often have control of your destiny.
I mean, this discussion has inevitably sort of focused on companies that are practicing drug discovery, but technology companies and support and reagent companies should not be ignored in this discussion. I mean, they can turn from very tiny companies into absolute monsters. I mean, Louis Sigma was a shoe manufacturer, and he got persuaded to sell ATP in the late 1940s because it wasn't available. Every biochemist had to make their own ATP. Well, the Sigma is now one of the largest 500 companies in the USA. The same can be said for companies like Invitrogen, Thermo Fisher, and here in Cambridge you have Abcam, which has a market cap of about £2 billion, and probably within a few years' time will be in the FTSE 100. So you shouldn't ignore these. There's massive support companies that is another avenue. And there you don't necessarily need an IP. You need to have a first-class product, first to market, and very good marketing.
Some more questions. That corner is very shy. No? All right. That young man with the white shirt.
Hi, thanks for the useful advice. My name is Nick Matthews. I'm a PhD student at the University of Manchester. I want to ask for some sort of practical advice that really relates to some issues that I think we had in our team for the Voice of Tomorrow competition. Is how do you, when you're setting up a company, how do you manage competing priorities when we're sort of doing PhDs, or spending the weekends trying to write papers that we've got to do to get the PhD, and then there's not 1,000 a day to also set up a company. So it's sort of, is it the kind of thing, should we wait until after the PhD and then set up and focus all our efforts on the same company we want to set up, or can you do it in parallel?
How do you manage your workload, guys?
Work long hours. No fun. Stay in the basement.
Well, perhaps I'll be slightly controversial. A colleague of Greg's, I won't name who it was, but I was talking to him about stepping down from a very significant role, and I said to him, "How do you feel about that?" And we had quite a long conversation about time management, and his conclusion was he was going to enjoy spending far more time on far fewer things. And I thought that was a very wise statement. And in the end, you have to decide what's important. As someone who has been notoriously unfocused, this is a very almost academic answer, because I've normally tried to do far too many things in my life. But I think if you want to do something substantive and difficult, then you have to focus your attention on it. And so that probably means that you have to make some choices. But the great thing about almost everyone in the audience is you have the most precious thing at your disposal, which is time. And time is the most precious asset that you have. So focusing your time on a small number of things, I actually think is at the heart of this.
Greg?
So just on your, I've never tried to set up a company when I was doing my PhD, so I can't really give you good advice on that, but my advice would be...
You're such a failure, Greg.
No, I know, I did it, was quite many years later, but, when I did set up a company, I had to work hard, 7 days a week for 7 years, right? So, and for the advantage of working with Australia was that actually we're out of time, so actually I could work in the evenings and actually get things done, and we could get experiments going in the day, and we would know what the answer was. So that was actually proved quite good. But what I'd recommend in the case of a PhD thesis is I notice with my own PhD students, they seem to think that someone will read their PhD thesis other than their examiner. Their mother won't read it, and their supervisor might not even read it. And, a PhD thesis just has to be good enough. No one's ever going to look at it and say, it's a scrappy job. They can say, is it good enough? Is the work in there good enough for the purposes of getting a PhD? So in your case, if this is what you're thinking about, what do? Get on with the company, but do the minimum necessary to get your PhD. Your supervisor won't thank me for that, but I think what is it you actually need to get a PhD? Focus on that and do the other stuff.
Clive, have you got that for the FT tomorrow? Greg Winter said nobody's going to read your PhD.
Greg is
just do the bare
correct.
minimum. Some more questions, please. Yes, over there.
Hi, actually I have a follow-up question on the topic of giving books to young entrepreneurs in biotechnology, which has been coming up a lot. And my question is it the right time that the entrepreneurial component should be added to graduate school education itself? I mean, people are studying for PhD, learning, becoming a good researcher, and then thinking of starting a company. But what if we can integrate the two into one resume and provide some PhD students a track? If they want to go to academia by starting a startup or to industry, that they can go on that track, and someone who wants to go for becoming a pure academic and give them flexibility of options during the PhD education itself.
Would you be happy for the PhD to be 5 years instead of 4 or 3 or?
Well, it is in some places.
Well, I think, you can't shortcut training. This is why we have MD, PhD sort of programs, etc.
I mean, you can we have a lot of PhD programs within industry, certainly within AstraZeneca MedImmune. We fund a lot of joint PhD programs, clinical fellowships, postdocs, and I think actually finding a way of having more exposure to a more industry environment through some of those programs is a really sort of good thing to do. And actually, I think, again, that's one of the gaps that's closed over the past sort of couple of decades. There's a lot more funding of PhD programs and actually some really nice joint schemes with a lot of the different types of funders to enable that. So I think to look for something like that to help oil the wheels of getting that industry understanding. Yeah.
So we've got 7 minutes, 54 seconds left. So I'm going to ask each of the panelists to give half a dozen words of advice to this young hundreds entrepreneurs, scientists, PhD students, MBA, lawyers, whatever, etc. Stephen, starting with you. 6 words.
6 words. Well, I think focus on what it is that you really want to do. Figuring out what you really want to do is at the heart of really everything, because if you start working on something that you don't really enjoy, you don't really believe in, then it's very unlikely to succeed. You don't have to have all the answers, but focusing on what it is you really want to achieve, whether or not that's making money, setting up a company, being an academic. Once you've figured that out, everything else becomes relatively easy. And I say that as someone who never really figured out what he wanted to do in the first place. So
I'm sure that was the longest 6 words. Okay. Greg?
Okay, make sure it's important and go for it.
There you go.
That's 6 words.
Focus, impact. Okay, Ray?
Build your network, like coming to a place like this. It's very, very important to build your network because you'll need it later in life.
Beautiful. Jane?
Be bold, really think about what it is that you want to do, and don't compromise on funding or ideas to get there. So be bold, and I would also say get as many different opinions as you can. You don't have to listen to any of them or take all of them on board, but make sure that you get diversity of views on what it is that you're doing and really ask for external critique. Yeah.
Wow, this is amazing. Focus, impact, network, bold. Phil, last word.
Don't try and decide things logically. Go with your gut feeling.
Yeah. I thought that was going to take 7 minutes. We got 5 minutes. So more
we can
questions.
do it again.
You're limited to 6 words.
Yeah.
More questions.
Thank you. So this is more like a question for you, Jane. We've heard a lot about the innovation in an academic setting, but how do you, as a big pharma company, MedImmune, stay innovative?
So, well, the first thing I'd say is I think that we have a view that if you do really good science, that will drive commercial success, and it's not the other way around. And I think that's been a step change, certainly within our company over the past sort of 5 or 7 years. The idea that you really understanding the mechanism of diseases and really getting to grips with fundamental biology is the way to innovate. And I think if you put that first, and you can then create differentiated products that really do make a difference. And last year we had 4 approvals for biologics, checkpoint inhibitors, anti-eosinophilic antibodies. You can really do something different. So I think that's the key. Don't think about the commercial environment too soon. Really think about the science. And the other thing is hire really good people. Really have a very robust process for assessing people, and make sure that there's a good team fit and that there's the right kind of attitude around wanting to innovate, wanting to disrupt. Hire mixed, diverse teams, actually, for all different ages, all different geographies. And I think that really gives you an element of disruption. And I think, again, we focused on that. So put science at the heart of what you do and really try and get a diverse workforce with some really innovative motivational drive to deliver on that. And it has worked. I mean, we've been, from the early days of being acquired by AstraZeneca, we set out a vision 10 years ago to deliver 1 new drug sustainably from 2016. And we already have started delivering on that. We will deliver on that. I have a very confident view of how our pipeline is innovative. And I think that's why. It's because we've looked at the disease rather than the commercial environment. So.
You're a great advert for the industry, Jane. So I think that was the first question from a young lady this afternoon, is that right? Maybe we could have some more questions from young ladies. Just here. No.
Hi, so I'm Katrina Nicole, so from AstraZeneca. My question
You don't work for Jane, do you?
Yeah. Was if you had the opportunity to start over your careers again, what would you do in today's industry?
Oh, wow. Okay, let's start right across. Stephen.
Oh, God, that's not fair. I would go into academia as I did last time.
Greg?
Yeah.
You'd go and work for GSK?
No, absolutely not, no. I would do something...
Now, hang on, I've got shares in GSK,
yeah,
Greg.
I would do something very similar to what I did last time, which I did accidentally, which was to go to sort of the top lab, which was the MRC laboratory of molecular biology, and work bloody hard. So I would find a really good lab and go there and then just throw myself into it and not worry about my pension or anything like that. Just go for it.
Have you been lucky, Greg?
No.
Right?
I would have liked to have been a physicist, but because of the nature where I come from, I was born in Egypt and all that stuff, I could never get into a nuclear reactor or anywhere like that, so it was quite dangerous. In those days they had severe clearance, and probably they still do today. But I think what I've done is probably the right thing, and if I were to do my life, I take the question further, if I start my life again all the way back, I would become an architect. Look at the beautiful structure here.
This has lasted a few hundred years?
Exactly.
Jane?
So when I started out, I took a decision that molecular biology was the most exciting thing around, and that's why I did a PhD in molecular biology, and that's why I ended up working in antibody engineering, actually. And I think I've been very privileged to have been exposed to massive opportunities in that field, and and I think that has been a really rewarding career decision. I was actually asked this question when we have an apprenticeship program. I was speaking to those guys around what would you do now, and I think I would just do the same again. I think at the minute CRISPR is the most exciting technique around. I would find the best lab, go and work on CRISPR, and really think about how you can apply gene editing in whatever. Don't be limited by your own imagination. So that's what I would do.
Thanks, James. Bill?
I've also been asked this question before. In fact, there was something, the Biochemical Society had something called 30 Years of the Colworth Medal, and they asked the same 5 questions to all the first 30 medalists, and all of them said something similar to the other members of the panel except me. And I said that if I were starting again now, I'd like to become an entrepreneur in the oil industry. Because I've become completely fascinated by the amazing technologies, how they can drill 2 miles below the surface of an ocean 2 miles deep. When there was this Gulf of Mexico disaster in the oil industry a few years ago, everyone was, giving BP a hard time because, they didn't fix it quick enough. I was simply amazed that they were able to fix it at all, because, they had to use these, midget submarines, by robot 2 miles below the surface of the sea in order to plug a very narrow pipeline with some cement. The technology is unbelievably impressive. So, yeah, I would become an entrepreneur in the oil industry.
So we've run out of time. So maybe if I could just leave you with a few words. So I was giving a talk in Harwell a few weeks ago. So Harwell's about 10 miles south of Oxford, and there's a lot of sort of engineering capabilities there. There's a synchrotron, etc. And at this, somebody asked me a question, and I said to them, Bill Gates has changed the world once, and he's changing the world for the second time. Zuckerberg has changed the world. Bezos has changed the world. Elon Musk has changed the world, or is changing the world. Where are those sorts of individuals in the UK, apart from Greg Winter? No, seriously. And I believe this country's got brilliant universities. We've got a wonderful track record in discovering new medicines. We've got great entrepreneurs. We've got great innovators, lots of talent. We've got a wonderful NHS that's ripe for exploitation in terms of research, etc. We have lots of funding in the UK. We have more funding than any European country for setting up biotechs, etc. The only country that I think that's got more is US, definitely, and probably China as well per head. But, I'd be interested in your thoughts. People in the UK who have changed the world. Thank you very much.